if you are sharing device and all of that, you don't necessarily have to be writing there in the chat. That's all right. Fine. If you want to write, it's okay. But I know some people will not have device, some will have to join others. But the most important thing for you is uh, have the benefit of understanding the material which is being taught you. In any case, it's already complex on its own. So if you happen not to attend the class, well, that is enough punishment for yourself. So just make sure that you have the best out of all the lectures that you have over the period. After all, it's not free, you paid for it. So you should make good use of what you have paid for. That's just a kind advice from me. Okay, so we start with gene regulation in eukaryotes, and the first thing we look at is two major processes that are involved in the regulation of genes in eukaryotes. Now, remember I mentioned when we were looking at the structure of DNA, we looked at the primary structure and the secondary structure. And we ended there. And I mentioned that actually you can have a tertiary, quaternary structure of DNA, but where we got to, it was just okay. The double helix structure of DNA was just okay. Now, what you need to know is that DNA molecules inside the nucleus of a eukaryotic cell are organized in the form of chromatins, which you find in the chromosomes. Now, what is the chromatin? So these are DNA molecules that are wound around proteins which we refer to as histones. Sometimes you see it being written as histone proteins. So it means that DNA molecules in a complex with some proteins. So you see it being depicted in this image here that you have. So the violet strands that you see, these are the DNA strands. And you see some teal color round divided into about like 4, because I mean to show that you can have those proteins it's a complex of, or you can have them as tetramers, like 4 subunits of proteins which are together with the DNA strands wound around them. Now you see that the DNA molecule is very compact in this form. It's a form of increasing the, you can say, surface area. I mean, it's a form of packing. It's a form of storing enough information, like more and more of information in a very small location. So it's just like you have let's say you have a long thread. So if you have a long thread and you have it long, just a long strand, it will occupy a very large space area. But if you have a reel that you roll it around it, you see that it becomes compact, and you can easily pack it in a very small area. It's similar with DNA molecule being wound around histone proteins, and actually this is the quaternary structure that we talk about, because you have this time around is a complex of histone proteins together with the DNA molecule packed together in chromatin, and this information is very compact together. Now, when you have the DNA molecule wound around the histone proteins this way, such that there is no access to the codes so that there will be the process of replication. I mean, this is a logical way you have to think that if you have something wound around, if you have the DNA molecule wound around a histone protein, then obviously you will not have access to certain portions, certain parts, the genes that you need to be replicated, the genes that you need to be transcribed onto a messenger RNA and to be translated into a protein will not even be accessible in the first place because they are so wound, so tight, packed together. So there's no way that you will be having access to those genes to have them transcribed and translated. And such compact form of DNA molecules are usually methylated. So you have the DNA molecule itself, with a methyl cap, that is CH3. And you also have the histone proteins themselves also being methylated. And in that form, the genes are inactive. The genes will be inactive in that form. It's a form of regulation of the gene. I think if you go back to the videos that I shared with you, think in the one on transcription, the two of them, I gave you two videos on transcription. You will see there is a process, there is a processing of RNA molecule, or I think DNA, after it gets synthesized. The new molecule gets some methyl cap on it. You also have some poly A tail, I think, if it is in the sense of messenger RNA. You have these caps at the ends of it. This is a form of protection of the genes. In that form, the gene is inactive. Now, you see also just below that, you have an active form of the gene. So here we see that it is depicted at the top here that there is a certain portion of the gene that is supposed to be expressed, and that portion is inaccessible. So it's written here, DNA inaccessible. Yes, it is inaccessible because it is compact together with the histone protein in that complex. Now you see it is being depicted below here when the gene becomes accessible. So now the gene is active because there is an unwinding of the DNA strand from the histone protein to expose it. So in this form, you can have the gene now getting transcribed and to be translated into a protein. And what causes that unwinding of the DNA molecule from the histone proteins to make it active or accessible for transcription, and then the messenger RNA getting translated? That is by the process of acetylation. So you see, acetylation is doing the opposite of methylation. So in other words, methylation is a negative regulation, whilst acetylation is a positive regulation. So going back to where we started from, you can see clearly that methylation is a negative regulation, whilst acetylation is a positive regulation, because methylation is preventing the gene from getting expressed, making the gene inaccessible, whilst acetylation is making the gene accessible to be expressed. So it is promoting the expression of the gene, and thus acetylation is a positive regulation. Now we see this process of acetylation, the activation of the gene, more in detail on the right-hand side, where it is being depicted more. So The structure you see below here is that of a histone, and that black lines around it representing the DNA strands that are wound around the histone protein. And we see that there is some lysine residue, lysine and amino acid residue also depicted in this model for you to see clearly that yes, these lysine residues, they have extra amine groups in them. All of this should tell you that in order for you to have a better understanding of this, you need to go back to your first year biological chemistry and go and look at the structure of lysine, the amino acid. When you were studying proteins, you looked at the structures of the amino acids. You can go and look at just the structure of lysine, and you see that it has an extra amine group in there. Now, that extra amine group, you can see, is having a positive charge on it. Then, you also remember that nucleic acids are made up of nucleotides. And these nucleotides are carrying negative charges on the phosphate group. So what it means is that if you have the lysine residues that are present in the proteins, that is the histone proteins, you have lysine present in those histone proteins carrying a positive charge, and the DNA molecule is carrying a negative charge on the phosphate group. Then you have a certain form of attraction between these two molecules, that is the histone protein and the DNA molecule. In that case, because of the attraction between them, you have a more compact complex together. Now, when the acetyl group, this is the acetyl group, which is 2 carbons, so you have a C double bond O with CH3. So this is the acetyl group, which is being shown here. When you see the acetyl group bound onto the lysine residue on this nitrogen, the nitrogen of the amine group, the extra amine group in the side chain of the lysine, you see that clearly the positive charge on that lysine group is lost. And if the positive charge on the lysine group is lost, obviously there will not be that attraction between the histone protein and the DNA molecule that is wound around it. By that, then you have the unwinding of the DNA from the histone group, from the histone proteins, so that the portion of the DNA, the gene that is to be transcribed, becomes accessible. That is the mechanism by which acetylation causes the activation of the gene. All right, so you can see that clearly. So I think if you just pay attention to the lysine, which is carrying a positive charge, and then by the end of this, let's call it a conjugation reaction. It's a conjugation of an acetyl group. Actually, this will be a group coming from acetic acid, which but in the body it will not be acetic acid, but it will come in the form of acetyl-CoA. So acetyl-CoA, which you also saw in the Krebs cycle. So acetyl-CoA gives out the acetyl group, it dissociates from the coenzyme A, and then the acetyl group now binds to that lysine, the lysine residue. Then you have that positive charge lost. Once the positive charge is lost, then there won't be that attraction between the DNA molecule and the histone proteins. And that will result in the unwinding of the DNA strands from the histone proteins. and that in turn results into the exposure of the gene or the accessibility of the gene to get transcribed. And once it gets transcribed into a messenger RNA, it will then get translated into the protein that it is coding for. All right, so that is a major mechanism of gene regulation in eukaryotes. The process of methylation uh, being the process by which the gene uh, is negatively regulated, and the process of acetylation by which the gene is uh, positively regulated. Now we look at different models of gene regulation in eukaryotes. So clearly you can see that there is an interaction of gene, which is a nucleic acid, and proteins, which is a form of a complex interaction between two different biomolecules, bringing about the regulation of genes in eukaryotes. Now, these can occur in different ways, and of course, what is being depicted here is not exhaustive in any way, but it is to give you a fair idea of the various things which happen as far as gene regulation is concerned in biological systems. So here, the first thing which is being depicted here is that you have a gene. So the strand you see here, gene 1, this gene can get activated by three different factors or three different proteins, which are 1, 2, and 3. The gene can get activated in that manner. What it means is that the gene will get activated when you have these three different proteins which are depicted, these three different proteins binding onto the gene at specific portions. Then the gene gets activated. These, can be referred to as transcription factors, or, the process which we just saw, acetylation, methylation. You saw there was some interaction between the DNA molecule itself and a histone protein. The enzymes which were involved really are not paying so much of attention to them. Those proteins not paying so much attention to them. But we are only seeing the different ways that interaction can occur between DNA molecules, or call it genes, that is nucleic acids, and proteins, which bring about activation or inactivation of genes. Now, the second thing which can happen is that you can have, let's say, these are also like three different proteins where you have the gene being activated by the interaction of a protein 1 and protein 3. But you see that in order for the activation to occur, there is a third protein that is numbered 4. That brings about some interaction between the 1 and the 3. You see that this protein 4 is not having a direct interaction with the DNA molecule itself, but it's having interaction with the two proteins that are having direct interaction with the gene. You see the same thing happening. You see what is happening with gene 1 is different. In gene 1, you see that the three different proteins are having direct interaction with the gene to cause its activation. In the second, you see only 2 proteins are having that direct interaction with the gene. The third protein, which is numbered number 4, is not having direct interaction with the gene, but it is causing an interaction between the first number 1 protein and the third. It means if the number 4 is not there, there will not be activation of the gene, even though it is not having a direct interaction with the gene. You can also have a case where you have, say, these proteins 1, 2, 3, just as they are here in gene 1, these proteins 1, 2, and 3 can be having interaction directly with the gene. But when you have another gene, which is depicted number 5, coming in to interact with the gene directly, it causes the dissociation of, say, one of the proteins, which is number 2, to leave that complex. I mean, once it leaves, it means the gene no longer gets activated. That is a form of negative regulation. And so the gene gets inactivated when you have number 5 coming in. You can see that number 5 in this case is acting as a repressor of the gene. It comes in and causes the dissociation of an activator away from the gene that is to be activated. But like I said, this is not the only models that you may encounter as far as gene regulation is concerned in eukaryotes. There are various complex processes, mechanisms of gene regulation. But this is to give you a fair idea that you can have various combinations of factors that are involved in either the activation or inactivation of genes in eukaryotes. Now, what you need to also understand is that, you see, when you look at the mechanism of action of enzymes catalyze biochemical reactions in such a way that it isn't that an enzyme, which is a large protein molecule, its whole surface is having interaction with a substrate on which they are acting, but you have a specific portion of the enzyme which is having that direct interaction with the substrate. And that specific portion of the enzyme we refer to as the active center of the enzyme. So you can have a large molecule, large protein molecule, but not that the whole surface of the molecule can have direct interaction with a substrate to be acting on it. The same way, we have just talked about proteins interacting with DNA molecules, proteins interacting with nucleic acids, forming complexes, which will result into either activation or inactivation. Now, the question you have to ask is, so which specific portion of these proteins will be having that interaction with the DNA molecule? It's just the same way as I talked about the interaction of enzymes, proteins with their substrate. There will be some, there are specific portions of these proteins that have direct interaction with the nucleic acids, with the DNA molecule, that gene, to get it activated. It is that portion that has that high affinity for the DNA molecule to have that interaction with it. And these interactions come in different forms, like hydrogen bonds, van der Waals forces. You have this interaction between the DNA molecule and the protein. With enzymes, we refer to these specific portions of the protein molecule that is interacting with the substrate as active center. In this case, the specific portion of the protein which is interacting with the DNA molecule, we refer to them as motifs. So when you read biochemistry books, you come across this word motif. It only means a specific portion, a specific part. And usually it's just a few amino acids that are in the whole protein or polypeptide structure that form that portion which we are referring to as motif that will have the interaction with the DNA molecule. Now, these motifs have been grouped into different categories. We have what we refer to as helix-turn-helix motifs, the zinc-finger, leucine, zipper, the helix-loop-helix motifs. And really, it's not anything complex about them. It is just about the structure, how they look. That is why they are called by these names. So we'll go ahead and look at the structure of these motifs. Not so much into looking at, the various parts, molecules, and the atoms and all of that are involved. No, we are only looking at the structure, how they look. That is why they are called like that. So, for instance, the helix-turn-helix. I mean, if you just, so the black lines which you see refer to the DNA molecule. And then you have the red or the magenta lines referring to the protein molecule. And that the specific portion of which that is having an interaction with the DNA molecule. And this helix-turn-helix is called so just because when you look at it closely, you realize that there is that form of a ribbon nature of a portion of that protein. And then you see there is one loop. And then right after the loop, you see that there is another helix structure. And that is why it's called a helix-turn-helix. So there is helix, and then there is a turn, then there is another helix. That is why they are called as such. Now, just a few minutes, I could realize that some people could easily, their mics are easily coming on and noise is coming in. So I want to make sure that nobody will be able to unmute themselves and start to make noise into the class. Okay, actually the settings are okay. The mic is not allowed, but I don't know. So that's technology. You can have these settings, all right, but sometimes some of these things can happen. Okay, so we can continue. Right, so you have the helix-turn-helix, and I mean an example of this is the lactose repressor. So remember, the lactose repressor is a protein. So if you go back and look at the lac operon carefully, you realize that there was a regulatory portion of the lac operon, which was labeled I. And from that I, that is coming from the word inhibitor, you have transcription of that portion into a messenger RNA that got translated into the repressor, which was a tetramer. So that repressor is a protein. And that repressor now goes to interact with the operator region of the lac operon, preventing it from getting expressed. So that is an interaction between a protein and a nucleic acid. So the lactose repressor has that structure, the helix-turn-helix motif. And usually you have this helix-turn motif being, composed of, say, 20 amino acids. I mentioned to you that you have a large molecule, and within it you have about a few amino acids which will form the motif that have the direct interaction with the nucleic acid. Now, you also remember that when we were talking about the activation of the lac operon by the complex after you have the repressor leaving the operator region. That is when the gene is activated, you have a complex of a catabolite activator protein and cyclic adenosine monophosphate. So, I mean, we are only building on what we have studied previously. So clearly, you have a protein and then another nucleic acid. This is a nucleotide, cyclic adenosine monophosphate, which bind together to form a complex binding onto the promoter region. Then that activates the gene to have the RNA polymerase coming to bind onto the promoter region, and the gene begins to get transcribed. That is specifically on the lac operon. So you can go and look back at it and you'll see that, yes, what we are referring to here is that the catabolite activator protein that is also having that interaction with the gene, the lac operon, is also having this structure of the helix-turn-helix. So, I mean, this is something which you just need to know that, yes, you have this motif, this protein structure, which has a direct interaction with nucleic acids, and not much into the various components that are inside this helix-turn-helix. No, we're not going into that. We are only looking at the visual structure. You just see how it looks, and you'll be able to tell that this is a helix-turn-helix structure. Similarly, we have what we refer to as the zinc finger. So, I mean, the zinc finger, for you to understand it well, think of the charger, the charger that we use in the car. There is this spot, the slot that you fix the charger into. Now the charger itself that you fit into the car, you see there is some sort of a lock at the sides of it, so that when you push the charger into the port in the car, it locks in that port, and then you can use it to charge. So you see how the sides bulge out, the tip of it, how the sides bulge out, and the tip of it, the structure of it looks just like the tip of the finger. It looks just like a finger. If you just raise your thumb against it, it looks as if like it is a finger. That is the reason why it's being referred to as the zinc finger motif. And the zinc finger motif, generally binds to the major groove of DNA, which is made up of about 5 base pairs in DNA. So these things you have done, the major groove, minor groove of DNA, you've done these things in first year. So there's no need going into these things so much. But the structure of the zinc finger is such that you have a zinc molecule, the zinc ion, being a central ion, that you have, say, the protein molecule, which is represented with this, the protein molecule, which is represented, part of it represented with the black lines, and then you have these the central zinc ion at the center, and along the strand of the protein, you have the amino acid cysteine at some portion. So the C is referring to cysteine, the amino acid cysteine, and two cysteine residues at different positions, having some disulfide bridge between them. And on the other side of the finger, you have the same structure. So 2 cysteine having some disulfide bridge between them, and on the other side, you have also 2 cysteine residues having a disulfide bridge between them. That together gives you the structure of the zinc finger motif. Or you can have zinc-finger motif also having on one side two cysteine residues having such disulfide interaction between them. Or you have one on the other side having histidine. So two histidine molecules also having such interaction between them, except that here it is not a disulfide bridge because, of course, we don't have uh, uh, sulfur in histidine, but rather you have a certain ring structure and you have some form of some hydrogen bonding between these two histidine molecules. So you can have such a structure, the zinc finger, which specifically uh, has a direct interaction with the major group of DNA molecules. And examples of these are like some steroid hormones, steroid hormone receptors, which act, or let's say they are a form of, they are receptors to some transcription factors. So this is one form of the motif, the structure of protein that can have a direct interaction with the DNA molecule. Now, the other structure is what is referred to as leucine zipper. So these two, when you look at it, it looks just like a zip. So you have these portions of proteins that are rich in the amino acid leucine. So you have the strand of protein, and then along the line you have a lot of leucine residues that are packed, and these form a structure looking just like a zip. That's why it's being called the leucine zipper, because it is looking, uh, just like a zip. You find this in what is being referred to as enhancer binding proteins. It's been seen, for instance, in some proteins that participate in the progression of cancer. For instance, there are these proteins which, at a point in the progression of cancer, even in the treatment of cancer with the phototherapy, they sort of develop they participate in the development of resistance to the UV radiation. Sort of there's a process referred to as apoptosis, which is you can call it cellular suicide. So a cell at a certain point in its life, it can trigger its own death. So seeing that for maybe the cell has sort of gotten some interaction with, it could be some infection from maybe a viral infection where maybe a virus infects the cell, and the cell noticing such an infection, triggers sort of an immune system. It triggers its own death, so it begins to break down so that it will not affect other cells. I mean, the biological systems are very protective, self-protective of themselves, in a way that a cell can actually cause its own death in order not to infect other cells. There is such mechanism, which is referred to as apoptosis. And in that process, you have proteins such as what is referred to as the force and John. These are very much seen in the progression of cancer. I mean, that's just by the way, just to have a certain practical application, to know that these things, it isn't that it is just theoretical things that like you just see the structure of, say, the leucine zipper and that's all. But these are things which are actually in medical applications. All right. If you are interested, you can go read more about it, uh, which is okay. You broaden your mind, even though you are not going to be examining on any of these things. Then you have the helix-loop-helix. So here too, just by looking at the structure, you can tell there is some helix at the beginning. You see a helix structure, and then there is some loop along the line, and then you go for another helix again. You see it on both sides. So you have the helix-loop-helix. These are two. It's let's call it's a two protein structure. So it's dimeric. So on both sides, just like with the leucine zipper. When you look at the leucine zipper, it's also dimeric. So two proteins that are in a complex acting together, and both of these proteins are having the leucine-rich residues at both sides that are having that structure looking like a zip. The same way you have the helix-loop-helix, two proteins forming a complex together. That is a dimer. It's a dimeric complex that is acting, that is having an interaction with the DNA molecule. Right. So that is to do with the protein motifs. Now, another form of gene regulation, which is in eukaryotes, is what is referred to as gene amplification. Now, in this case, gene amplification here is not referring to the technology of the PCR, where you want to you have a reproduction of DNA molecules. That's not what is being referred to. But in this case, what gene amplification refers to is the fact that there are certain drugs, just like okay, so there's an example here which is methotrexate. Methotrexate is a drug for cancer, but when it's being administered over a very long period of time, the body begins to develop some resistance to the drug. Some drug resistance begin to be developed, and the mechanism of that is such that the cancer cells uh, now get sort of adapted to the environment of the methotrexate drug, because they notice now the methotrexate drug is causing their death, and so they begin to transcribe them more. So there will be an elevation of transcription, so that there will be an increase in gene expression, so that even if they were dying, they will now be in larger quantities, such that it will compensate for the death of it will compensate for their death. So no matter what, the cancer cells will begin to be there. So some of them will die, but because now they have increased their process of reproduction, the process of transcription, they will begin to reproduce them more. That's like reproducing, over reproducing. Of course, a tumor only means that you are having an elevated form of proliferation of the cells, growing of the cells, and so yes, the drug is being administered, all right, but the increase in the proliferation of the cells will overweigh the action of the drug. That form of gene regulation is what is being referred to here as gene amplification. It is unintended. The drug is not introduced into the system to go and cause the gene to get overexpressed, but the body, those cells, the cancer cells themselves, begin to develop such a resistance, and have specifically the transcription of the genes that code for the enzyme hydrofolate reductase, which is responsible for the metabolism of folates, tetrahydrofolates. These we refer to them commonly as folic acids, and these folic acids are responsible in various reactions which are needed for the development of, if it was in fetus, the neural tubules, in other words, the brain. And in fact, generally, the growth of cells, the proliferation of the cells, you have these folates acting. I mean, that's the reason why folic acid is prescribed a lot for say, pregnant women when the fetus is developing, so that you can have enough of it to help in the development of the brain, of primarily for the brain, even though it is generally for the whole body of the fetus, but primarily for the development of the brain. The lack of it will result into some maldevelopment of the neural tubes, which is the brain. All right, then you also have in eukaryotes, I mean, you can have what you refer to as recombinant DNA. I mean, rearrangement of genes. The body is able to have the biological systems, the eukaryotic cells have this ability to rearrange the genes, so what it means is that if you have a certain sequence of genes that is giving you a specific protein, and the system is able to rearrange this sequence in a way to give you a different protein altogether. Such a way is also a form of gene regulation, so that you have some diverse proteins which will be having different functions in such a biological system. That is referred to as some form of gene regulation, gene rearrangements. Many of the biochemistry books, you see it being referred to as gene recombination. So you see it being depicted here that yes, there's some portion here, the red portion, the blue portion, and then you have the black portion, different sections of the DNA molecule that are coding for different proteins. And then you see that there is a rearrangement such that you have some three of the, not specifically three nucleotides, but to show that there is some specific portion of it which is combined with a certain portion of the blue and then a smaller portion of the black. And you see such rearrangements along the line to give you different forms of genes. And then you now have a new protein, which is this is being depicted for an immunoglobulin. So an immunoglobulin, in other words, is a white blood cell. It's an immune cell. So this is just a model to show how genes can get rearranged, get combined in different ways to give you different forms of proteins. All right, so you see the gene, first of all, there is that rearrangement of it before it gets transcribed into a messenger RNA. And then from that messenger RNA, you have the protein, which is an immunoglobulin. All right. Now, when you watched the video on transcription, I think the long version of the transcription, you saw that after the messenger RNA was synthesized, that is the process of transcription, there was a certain protection that there was a processing of the messenger RNA. So then you have some poly-A tail. There was a poly-A tail. That is the nucleotide adenosine monophosphate attached to the 3' end. Then you also had, I think, some methyl cap also attached to the 5' end. That is a form of protection of the messenger RNA. In fact, the capping of the ends of the messenger RNA actually makes the gene inactive, so that it is only when the gene is to be expressed that these caps, get activated. They get CZ removed, and then you have the gene getting expressed. That is the coding region of the gene which is to be expressed, is what is taken out to get expressed. So that form of processing of the messenger RNA is also seen as a form of gene regulation, so that when those caps are there, the gene will not be expressed. It is when the gene is supposed to be expressed, then you can have them. Or in other words, so it makes the gene sort of stable, so that it is only when it is to be expressed that then the gene gets activated. Then you can have the transcription of this portion, the coding region of the messenger RNA into the translation of that coding region of the messenger RNA into its corresponding protein, what it is coding for. Right. Well, another way is that if the caps are removed, the gene gene is also exposed to be easily degraded, to easily undergo degradation. Once the gene is broken down, is degraded, it means that there will not be any form of expression of it. So it's also seen in that way. Right. Now, apart from all of this, you can also have what is referred to as epigenetic regulation, which means external factors that can affect the regulation of genes in biological systems. Some foods, for instance, so what people eat, what you eat can affect how genes get expressed. I think I made the example of the when I was talking about inducible genes, when we were looking at inducible genes, I mentioned the fact that these genes will get expressed only in the presence of the specific substrates. If a substrate is present that is supposed to be metabolized, then there must be the enzymes that will participate in the metabolism of such substrate, and so then those substrates actually activate the expression of the genes into the proteins that are required in their metabolism. So in that case, a diet, for instance, is a substrate that requires metabolism, and in their presence, you will have some genes being expressed to produce enzymes to break them down. Some other things like long-term disease. We mentioned. We talk of some examples with cancer. So some drug, methotrexate, can be causing some form of gene regulation. Sometimes, even apart from the things you take in, it can even be the sort of environment in which you are, the noise. That it can even be noise or some sort of irritation in the environment, and the body begins to react. So, I mean, look at it this way: different situations cause different moods in people, and what regulates those moods? It's hormones. So hormones will be secreted into the circulatory system, which will be triggering certain moods depending on the situation in which the person finds himself. So these are epigenetic factors, so are external factors that are causing the secretion of a particular hormone into the system. And that hormone which is being secreted, remember, is a protein, which means that protein will be synthesized, starting from that process of replication, uh, transcription, translation, so that in a particular sort of a situation, especially when it is a prolonged situation, you have certain hormone being secreted. Either you are going to be happy, or you are going to be sad, or you are going to develop some sort of not a mental disorder per se, but some sort of maybe a hormonal imbalance, for instance. So you begin to, secrete hormones at a high level or not at all, depending on situations which you find yourself. So these things are what you refer to as epigenetic regulation of the gene in eukaryotic cells. So I think at this point I will just open the floor so that if you have anything to say, at least we have some 20 minutes. I think somebody has written. Let me check the chat. See. Okay. He just wrote his name with his student ID. So the floor is open if you have any interesting contribution to give us. Say some of you have had some experience working in some places, or even some personal experience you can share with us. If you have any questions, I will attempt to give an answer meaningfully to it. So the floor is open. If you have anything to say, you just raise your hand. I will allow you to unmute yourself and you can speak. Well, nobody is saying anything. Does it mean everything is clear? Everybody understands everything? Okay, somebody. Somebody is saying, is this lecture on YouTube too? Maybe not. But I keep updating, so maybe this one I can put it there, so you can always have access to it. What I try to do is after every lecture, I put the video there on the link, so that anytime at all, anybody, you can go have access to it and make use of it. So I'm sure this one, when we finish, I will just put it there and send a link to the class rep, so you can look at it. Any other comment, question, anything not clear? It appears this is a very, very smart class, so everything is just clear to everybody. So okay, somebody is asking what is the name of the channel. So let me write it. I mean, you can go to this is This is oh, let me see. I think I should first write handle before I write it, because when I want to type the handle, it is seeing it as if I want to tag somebody. Maybe let me put it in inverted commas and say same. Well, but okay, I think what I should just do is get you the link, of the channel so that you can go there. I tried typing in the handle over there, but when I tried typing in, it is seeing it as if I want to tag any of you. And once the name doesn't come, the message tool doesn't go through. So I'm sending you the link to the channel so that, okay. So I've sent the link so you can see it. Okay, so if nobody has anything to say, nobody wants to ask any question or make any contribution, then I'll propose that we bring our meeting to a close till we meet the next time. I hope that okay that today, okay, the next Friday is going to be a holiday, that is 6th. So which means it is after the holiday that we can meet. But well, that will depend on you. If you wish that we have class, we can have class online. And after all, it can be a holiday, but online you are not going anywhere. So you can discuss among yourselves. If you decide that you want to have a class on Friday, we'll have it online. And then after all, it will only serve as a good purpose. If we are able to go through everything that we are supposed to go through, you have a longer time, you have enough time to do revisions and all of that. All right, so I propose that we bring the class to a close. Any secondment, you can raise your hand to second the motion, and then we close. Owusu Godfred is the first person to second. And Livingston Ejiro. All right, then I will say goodbye to you now, and have a nice time until we meet again. Bye for now.